Okay, so thank you so much to everyone for coming. And today we will have two lectures from Laura Zavotaro and from Kunju. And I will begin by introducing Laura, who will be giving her lecture first. Laura is a graduate with a master's degree from the Faculty of Agriculture of the University of Turin, and with a PhD degree from the Department of Agricultural Science and Technology of the University of Turin. She is currently an associate professor at the Department of Sustainable at the Department of Veterinary Sciences at the University of Torino, and her main research fields are long-term effects of nitrogen fertilization on soil organic matter and nitrogen leaching. She is also a member of the General Council of the Faculty of Agricultural Sciences and Technology, the Departmental Council, the Curriculum Council, and the Veterinary Degree Council. And her long-term research as a managed focuses on soil organic matter turnover and feed, livestock manure management, quantification of nutrient losses and farming systems related to agronomic techniques, and soils on farm assessment of agricultural manure management practices and soil hydrology. She has been involved in recent years in several EU Horizon projects, such as TUdi, FertiCycle, Super-G, and others. And she has published extensively in high-impact journals such as Nature, Ecology, and Evolution, European Journal of Agronomy, Journal of Clean Production, Agricultural, Ecosystem and Environment, European Journal of Soil Science, Soil Use and Management, and more, with a total of 60 journal articles, 40 international conference papers, and 35 national conference papers. She will be presenting today on the topic of soil nutrient budgets and manured cropping systems, evidence from a long-term experiment.
Thank you very much, Liliana, for this long introduction with several details. And thank you for being here. I hope that today's speech will not be too boring for you. So I will start sharing my screen. I hope you can see the slides. Can you?
Yes.
Okay, great. Okay, so today's speech from my side is about a long-term experiment that we manage here, and on the techniques that we have used a lot to explain and to interpret data based on the nutrient budget. So I would like to show you some examples on how this technique can be used in interpreting data from long-term experiments. The context in which we move is regards the European situation of soil quality that is not satisfactory, and this is one of the reasons why the TUdi project was financed. But we are also not happy about the quality of air and groundwater quality. Then we have a problem regarding legislation. The legislation that is moving towards a lower use of chemical fertilizers due to the fact that as a response to this unsatisfactory quality of the environment. The reduction in fertilizer use is because we want to increase the efficiency. That means that we want to produce more with less, in particular with less inputs from external sources. This should protect the environment, but should also protect the primary resources that we are using, in particular to produce some of the chemical fertilizers, for example, phosphorus, but also potassium. Another important objective of modern agriculture in Europe is to increase circularity. That means to change from a linear model of economy to a model that promotes the reuse and recycling of or everything that is produced within within each process. With this context, well, we have to find new solutions to modern problems of agriculture. And long-term experiments are a possible way to answer this kind of questions that we are posing ourselves. Long-term experiments have good qualities that sometimes make them better than short-term experiments. The first important thing is that they are already there, and they are there for several years. As we know that changes in soil are very slow, it's difficult to detect any change in short-term experiments, while long-term experiments can do that. Then another good thing is that while an experiment is running for several years, we can exploit also previous data, but also reinterpret data that were taken for other purposes. Some long-term experiments were financed to answer some questions, but now we can use them to answer new questions posed by modern agriculture. Long-term experiments are several across Europe, and the fact that we can network network with other long-term experiments in Europe and all around the world make any observation more robust and interesting. Then, as this kind of experiment lasts for several years, people around the place where they are located know them very well and know that they are a source of information where they can be inspired for improving their knowledge regarding agricultural problems. So they can be used as reference points in improving knowledge about agriculture. We think that long-term experiments are like good wine, so the longer they stay there, the more valuable they are. Today, I will show you how a long-term experiment can answer new questions, despite it was set up to answer different questions. So we can transform it in order to be able to respond to new challenges in agriculture. And I will also show you some applications of the mass balance to answer some of the experimental questions that we can answer using data from the long-term experiment I will show you. And the overall context is that good practice can improve soil health. So you will also get some information regarding the importance of incorporating crop residues and of manuring as means to improve soil health. This is the passport. So sorry, I was trying to. Okay, this is better. This is the passport, the ID card of the long-term experiment that we manage. It is called Tetto Frati, that's the name of the place where it is. And it is owned by the University of Torino, which is in the northwest of Italy. And in particular in the northwest of a long valley, that is the Po Valley, which is a flat area that occupies most of the northern part of Italy. It was started in 1992, although sometimes we dropped the first 2 years as they were more influenced by the previous management to have more reliable data regarding the effect of the treatments that I will show you. The soil is deep, it's calcareous, and it has a loamy texture. The experimental design is a randomized experiment with blocks, three replicated blocks. And the different cropping systems are compared in a in a design that involves 38 treatments. So the number of treatments is very high. We have 4 main cropping systems that are compared. There are compared at 9 fertilization levels. And all these fertilization levels are based on nitrogen fertilization, because the main objective for that gave the start of this long-term experiment was the nitrates directly. So the study on how nitrogen fertilization affects the groundwater quality and through leaching. We have different cropping systems that we will meet today during my speech. There are maize for silage, so it's a maize that is entirely harvested. Then we have maize for grain, where grain only is harvested and residues are incorporated into the soil. Then you have a special cropping system that we have sometimes here in Italy. There is a double cropping system. That means that we have 2 crops in 1 year. Italian ryegrass for during winter and maize for silage during summer. Then we have some rotations, and in particular a rotation with 3 or 4 years of grass, and then 3 or 4 years of maize for silage. These are all typical of this area of the Po Plain. Then we also have other fertilization, other treatments that involve only 1 fertilization levels. That is a rotation with lucerne. That's similar to the rotation with grass, but with lucerne instead. And a treatment that was set aside, so no cultivation in the first years, but then it was converted to permanent grassland. I will not show data on that today. The 9 fertilization levels include a zero nitrogen control, then mineral treatments where nitrogen is supplied as urea. Urea is not forbidden in Italy, while it is in other countries in Europe. And we have 4 doses that I have changed a little bit during time. But the main treatments that were that include the focus of the long-term experiment are bovine slurry and farmyard manure. They are distributed at 2 doses, and the 2 doses have changed a little bit in time. So I will show you some results from 3 different periods where manures were distributed different amounts and with different criteria. In time, we have reduced the amount of nitrogen supplied with this kind of manures to adapt to a change in legislation that is wants to reduce the amount of fertilizers, both manures and mineral fertilizers supplied to crops. This is the kind of data we can draw from this long-term experiment. You have the 3 periods with changing amounts, and the response curve to nitrogen dose, and the nitrogen dose is on the horizontal axis, while on the vertical axis you can find the dry matter yield of one of the systems that is maize for silage, so totally harvested maize. You have the urea response curve in gray, and manures that are compared to it. As you can see, in the first period, the farmyard manure was supplied at high doses, higher than slurry, and the efficiency of manure was very similar to that of urea. So it seems that supplying crops with manures, different types of manures, had the same effect of supplying the same crop with mineral fertilizers. In the second period, we changed the doses, and it seems that as the low doses, that was 170 kilograms of nitrogen per hectare, was very similar to the efficiency of that we could obtain with urea, while at the high doses, that was 340 kilograms, well, we could obtain a higher yield than urea. In the third period, here we have 12 years of another management. We can compare directly the amount of nitrogen supplied with mineral fertilizers and with manures, and the efficiency seems to be a little bit lower than urea at the low dose, but a little bit higher than urea at the high dose, that was 250 kilograms of nitrogen. So these are the data that are a little bit variable. And to understand if this situation with a high efficiency of manures is normal, we compare it with other long-term experiments that we found in Europe. This was a review paper that was developed in a previous European project that was called CATC, where we collected 80 long-term experiments in Europe that you can see here on this map. And we wanted to compare the manure fertilization with mineral fertilization at a similar nitrogen rate. So we found 80 long-term experiments that respected this objective. We wanted to find out which were the main factors that influenced the response of manures compared to mineral. And we explored soil texture, climate, but also the age of the experiment, tillage depth, and minerals supplement to manure. But today I will show you only data regarding soil texture and climate types on a limited set of indicators. So I will show you data regarding two crop indicators that were yield and nitrogen uptake. And as you can see, both in farm manure and slurry, the yield was lower than that was obtained with mineral fertilizers. And the difference was about 5% regarding farm manure to 10 and 15% regarding slurry, bovine slurry. Then we also calculated some nitrogen use efficiency indicators that were calculated differently, and the difference between manures and mineral fertilizers was instead higher using if we looked at this kind of efficiency indicators. Then we had three indicators of soil health, and that were soil organic carbon, soil organic nitrogen, soil total nitrogen as it is normally called, and microbial biomass. And in this case, instead, the effect of manuring was positive on all the three indicators, although there was a lower effect of bovine slurry, and there were no sufficient data regarding microbial biomass and slurry. So this was the context, and we were expecting a lower efficiency and a lower crop yield in manure situation. So why did we instead had so high efficiency in our system? Well, partly the answer is can be found if we look at the factors that influence the response. In particular, if we look at climate, we can see that there was a positive effect of climate on the response of farm manure and slurry, and in southern Europe countries, so in southern Europe climate types, there was a general increase in yield due to manuring compared to mineral fertilization. While it was different, the situation in other countries that were more humid and cold. And this was observed in yield, but it was not significant in nitrogen uptake by the plant. If we look at soil texture, well, again, we had a positive, a significant effect of the soil texture on the response to manuring. And in light texture soil, the effect, as the tetraphytic soil is, the effect was more positive than in medium texture soil. So again, we can find the explanation that were different from the general overview based on the fact that we were in climate and the soil condition that favored the a higher efficiency of manuring. If we look at soil organic matter content, in particular divided in soil organic carbon and soil total nitrogen, well, there was a positive, significant effect of manuring due to climate. And in southern countries, the effect of farmyard manure was even higher than that was found in other type of countries. And this was also observed in regarding soil total nitrogen, despite the statistical model that we use detecting no significant differences. Regarding soil texture, light texture soil had again a positive effect of farmyard manure, while the difference was not as significant with slurry. If we look at efficiency indicators, well, the situation was more complex, as based on the different formulas that could be used to evidence the nitrogen use efficiency. Well, the differences were significant or not significant. So the situation was a little bit more difficult to interpret regarding the different ways that we have to intend the nitrogen use efficiency. This was the context in European countries, in other European long-term experiments. Now, let's go back to the long-term experiments of Tetto Frati, and let's first introduce what we call the mass balance. The mass balance is the difference between the input and the output, so the input to a system and the output to the same system. And it is somehow related to the concept of efficiency, where the efficiency can be calculated using different formulas, as I said before. But in general, it can be interpreted as the ratio between output and input. The mass balance is a way to interpret data, and can be used to forecast changes within the system, as if the input is higher than the output, it means that there are losses, or that the system is increasing in its nutrient content. The problem is that it is difficult to quantify all entries or voices as inputs, but also in the outputs. For instance, if the system is the field, well, the inputs can be regarded as the fertilizers, the manures, and sometimes also the previous supplies to manures is also intended as an input, sometimes that is mineralized later than expected from manuring. But all nitrogen, I will in particular I will talk about nitrogen with only a few words about phosphorus in today's speech. Then irrigation water can bring some nutrients, in particular some nitrogen that is dissolved in irrigation water. But also seeds contain nutrients, in particular some nitrogen. Nitrogen fixation is a sort of source of nitrogen. And the residues of the previous crop are also another source of nitrogen that will mineralize for the crop in the year where we are calculating this nutrient balance. Atmospheric deposition can bring nitrogen and also limited quantities of phosphorus depending on the area. And the soil organic matter can provide nitrogen to the crop. As outputs to the system, there is the crop uptake and crop removal with yield, but also ammonia volatilization on the products of nitrogen denitrification, but also losses as nitrate leaching. Runoff can deplete the soil surface from nutrients, in particular some nitrogen, but in particular phosphorus, linked to the soil particles. And another way that make nitrogen disappear from the system is an immobilization, as soil organic matter. So it's the same opposite process of soil mineralization. These voices these entries are mainly thought about nitrogen, but some of them are also common to other nutrients, particularly phosphorus. So the mass balance can be applied to nitrogen fertilization. This is one of the possible ways that nitrogen balance and nutrient balance in general can be used in practice. Using the mass balance approach to calculate crop requirements is quite common all throughout Europe, and I would say in most cases also in other countries in the world. So the crop requirement is calculated a priori, so before the crop is set, based on general averages of the situation where the crop is cultivated. However, using this approach means that we know how to quantify all the answers that I showed you before. And to make this calculation easy to be used by farmers, we also need to estimate or to predict all entries using simple formulas. Unfortunately, this is not the case for most of the entries. In particular, losses cannot be predicted easily using formulas that help us to quantify all the entries. So every country that use this approach based on mass balance, well, has adopted a simplification to predict the crop requirements based on a simplification of the formulas, and in particular, one common way used in Europe to simplify the formula and to not to require a quantification of nutrient losses is to use another approach. Instead of quantifying losses, we introduce a efficiency coefficient that is applied to fertilizer, in particular to mineral fertilizer, but also to manure. And several countries in Europe use this approach to quantify the efficiency or inefficiency of fertilization in a context of quantifying the nutrient requirements of crops. So there are several papers that were published trying to quantify the efficiency coefficient of manures, and that is also what we have done. So I will show the results. A mention to the TUdi application that we have, APP that we have developed in our project. We have calculated the fertilizer requirements not based on the efficiency coefficient, but based on another approach. But again, based on the mass balance. So the mass balance was also used in the APP to calculate the mineral fertilizer requirement that you can see here in red, based on the mass balance between inputs that are here with this gray color, and outputs that are here with this yellow color. So the mineral fertilizer requirement are calculated based on the crop uptake and nitrogen in manures and nitrogen derived from soil organic matter mineralization, on previous crop residues, on previous crop manures that are mineralized in the following years, or in nitrogen fixation, nitrogen biological fixation, nitrogen contained in irrigation water or coming from atmospheric deposition, and also denitrification losses and volatilization losses as ammonia are quantified. So we do not use the approach that instead I will show you right now. Because several countries in Europe instead use the efficiency coefficient approach, and also the Italian legislation, the Italian way to calculate the fertilizer requirement uses this efficiency coefficient approach. So this is the formula that is quite common also in other countries in Europe. Something changes, but I will show you the Italian one that we use. And it's also set by the legislation on how the nutrient requirements should be calculated and to to adapt the fertilization amount to crop. So the mineral fertilizers are calculated as a difference using this equation, where again on your left you have the positive entries, and on your right you have the negative entries. So as you can see, there is no losses because the losses are included as the inefficiency of fertilization. Inefficiency of mineral fertilizer, but normally we use the default values of 1 for efficiency of mineral fertilizer. That means that mineral fertilizers seem that they are not subjected to losses. It is let's say, a political approach to fertilization. But instead we calculate an efficiency coefficient for organic fertilization and for manures in particular, and the range in our case depend on the type of manure, but also on the soil, and it ranges from about 25% to 85%, so a wide range of efficiency coefficient. Then we also calculate the residual effect of farmyard manure, and we calculate the effect of previous crop residues that can be positive or negative depending on the C/N ratio, and sometimes only the effect of previous grassland is calculated as positive. Then we have the supply, in particular of nitrogen and soil organic matter mineralization based on the soil organic matter content. Atmospheric deposition that range from 30 to 20 kilograms of nitrogen per hectare, and the nitrogen fixation by legumes. Then we have the removal as the only negative entry of this equation. So what we have done in the long-term experiment is to try to quantify all the entries mentioned here to support the legislation and to support farmers who want to use the coefficients to calculate the nutrient requirements by the crop. And I will show you almost some of these approaches that were all calculated based on the mass balance and based on the comparison of different treatments. As we have a lot of treatments, we can use them to calculate some of the effects of, in particular, previous crop residues. Previous crop residue, if the previous crop was a grassland or a legume ley, so a temporary lays, but also manure efficiency coefficient, and something about the mineralization of soil organic matter, and I will also show you data regarding the soil available phosphorus. We can use the TUdi long-term experiments to derive all this, but the problem and the limitation of this approach is that we have only one soil and one crop, that's maize, and only 2 manure types. So we can contribute to the knowledge, but not solve every problem regarding the quantification of those coefficients. First of all, the residues of the previous crops. The previous crop here was maize. We compared maize for grain, where crop residues were incorporated in the soil, with maize for silage, where residues were instead not incorporated, as the whole crop is harvested every year. Based on the comparison between these two systems on several years, here you can see an average of several years, from 2007 to 2014, as this paper was published in 2016, where we calculated that maize residues contained a lot of nitrogen, and about 100 kilograms of nitrogen, but the C/N ratio was quite high. And we found out that that the plant uptake was not influenced by the incorporation or not of the residues of the previous crop. But some positive effect of incorporating residue was seen in low fertilized treatments. So there was no positive effect of this mineralization of soil of crop residues. But instead, there was a positive effect of incorporating residues on the soil organic matter stock, and in particular on soil nitrogen. We found a good increase in soil total nitrogen. If the crop residues are not a good source of nitrogen for the following crop, well, they are a good source of nitrogen for the soil. Here you have the results from another paper that was more recently published, and you can see that crop residues that are here in this part of the graph, so on the right part of each of the two graphs proposed here, crop residue incorporation provided an increase in soil organic matter content, and the soil organic matter content regarded all forms of soil organic matter, so both the particulate organic matter, but also the organic matter that is in strict connection with mineral, in particular with clay, so mineral-associated organic matter. The increase was in all forms. Incorporation caused an increase in soil organic matter content, and it was, but there was a higher effect of manures than of crop residues, despite the amount of carbon that was contained in crop residues was really high. Crop residue incorporation caused a 16% increase in soil organic matter compared with removal, while the increase of farmyard manure was 30%, and the increase of with the fertilization with slurry was of the same order of magnitude as crop residue incorporation. So crop residues increase the soil organic matter content to a larger extent than what they contribute, and then their contribution to crop nutrition. What happens if the previous crop is a grassland? Well, we had two cases. We could compare the effect of incorporation of a grassland and also of a legume that is lucerne, sometimes called alfalfa in American English, but lucerne is the English term for alfalfa. So we obtained this result. The incorporation of rotational grassland residues influenced the following maize crop by introducing more nitrogen to the system, but this increase was only 27 kilograms as an average over the 2 years after grass plowing. So the contribution to the crop nutrition was not limited to the first year, but sometimes more in the second year after plowing. And this positive effect of incorporating the residues of a grassland was higher in lower fertilizer plots than in higher fertilized plots. But the effect of legume was the same of incorporating a grassland. So there was no legume effect that was unexpected that was unexpected, because normally instead other literature says that the effect of incorporating legume residues is higher than the effect of incorporating a grass. Again, we can use the long-term experiment to quantify the manure efficiency. The effect of manuring can be calculated using different formulas, and here you can see two different formulas used to calculate the nutrient use efficiency. Here in the first graph, the nutrient use efficiency is calculated as the simple ratio between removal and fertilizer supply. And the variability you can see here in this graph regards the different cropping systems, but also the different type of manures or mineral fertilizer that are all here in this graph. And as you can see, and as expected, the efficiency of fertilization changes also with not only with the type of fertilizer, but in particular with the dose. And the efficiency is lower if the dose is higher. But the overall variability of this coefficient is really high. And as you can see, can range from a number that is lower than 1 to a range to a number that is higher than 1. That is not really expected. it means that the efficiency, that the plant could take up more nitrogen than the amount that was supplied to it. If instead we use another way to express the efficiency coefficient that used also the unfertilized plot for its calculation, and if we refer it to the mineral, so to the urea treatments, compared to mineral, we can see that manure an effect that again is can be lower than manure than mineral fertilizer. Sorry, in particular at low amounts of manuring, but when the amount of manure is higher than a certain threshold, the efficiency of manure can be even higher than urea. So we have numbers that are higher than one here. It means that manure can behave much better than urea itself. So the efficiency can be even higher, but the problem is that this efficiency coefficient is difficult to calculate, and it's difficult to express as a single number because it changes with the crop, it changes with the manure type, it changes with the dose, and it is also a high variability from year to year. There is not a big here, as here we can only see averages. Another factor that we try to quantify that is based again in the mass balance equation to calculate crop nutrient requirements is the contribution of soil organic matter to the crop nutrition through the mineralization of soil organic matter itself. Here you can see the main result of a paper that was very recently published by Octavian, who's a PhD student working on the TUdi project. And you can see how the mineralized nitrogen, as it was estimated by the zero nitrogen plot, changes based on the year, so change in time, and change also based on the cropping system that was used to estimate it. And there are the different markers here from year to year. As you can see, there is a trend in reducing this contribution, this mineralization from soil organic matter. And but the trend is not the only source of variation, as there is some pattern that changes from year to year. Okay, the first news is that mineralization of soil organic matter provided about 100 kilograms of nitrogen every year to the crop. But this trend, there was a decreasing trend that was caused probably by a change both in climate and in the type of organic matter that provided this mineralization. So the main factors that influence this variability that we observed in soil organic matter mineralization was partly due to climatic variables, in particular the amount of rainfall, amount of water that was available for mineralization. That was the sum of natural rainfall plus irrigation. But also temperature increased while time passed. And this variability of temperature was in the statistical model that we used, was absorbed by another change that we observed. That was the amount of soil organic matter that was decreased in time. So rainfall irrigation, but soil organic matter amount and also the quality of soil organic matter itself, expressed as C/N ratio, explained part of the variability that we observed in this trend. but the amount of variability that was explained using these three main factors was only less than 20%. There were other factors that we could not interpret and to explain this variability in soil organic matter mineralization. In fact, if we included random factors such as the year-to-year variability and the cropping system, as we used the three different cropping systems for this analysis, well, we increased the variability explained to 52%. Another chapter regards instead the crop nutrient requirements, but not nitrogen, as we will also say something about phosphorus. The long-term experiments of Tettofrati was not designed to study phosphorus, but we could use it to study in particular the legacy effect. So the fact that different treatments after long-term manuring changed the initial available phosphorus content. So we had different plots with a different initial soil available phosphorus content. We had here we studied the effect of adding phosphorus at sowing, so together with the seed, close to the seed, based on the initial content of soil available phosphorus. In a soil that was poor in phosphorus, well, the effect of supplying phosphorus close to the seed had an important effect. There's the dark colored line here, dark green line, compared to not giving any phosphorus at sowing. So in a poor soil, the effect of this extra P supplied at sowing was positive. But instead, in a slurried and in a manure system where the soil was rich in available phosphorus, the factor of distributing some phosphorus at sowing was positive only in the first phases of the growth cycle of the crop, but at flowering there was no effect at all. So if the crop was a little bit different, as the crop was a little bit more developed in where the certain amount of P was supplied at sowing, where at flowering there was no difference between the plots that received extra P and the plots that did not receive extra P, based on the previous history of these plots. We could also use the phosphorus data, phosphorus available data of Tettofrati, together with other long-term experiments that we had in the neighborhood, to interpret what's happened to increasing fertilization and decreasing fertilization. We used again the phosphorus balance as a way to interpret data. So we had positive data of soil phosphorus balance where the fertilization was higher than crop removal. So, and this is what you can see in the right hand of this graph. While the negative numbers here on the left hand of this graph represent situations when the fertilization was suspended or was lower than crop removal. So we could accumulate a negative balance from year to year. And this long-term accumulation of negative balances was used as the interpretation of the changes we could observe in ozone phosphorus that was here in the vertical axis. The different lines here differentiate not only different soils, but also different matrices, so different sources of phosphorus used as different types of fertilizers. This, we could express all these different situations using an exponential equation as the one you can see here. And we had three parameters that differentiated these different situations. Two parameters were linked to the soil, and one parameter, D, was instead linked to the type of source, type of fertilizer. And what we found is that the C to P ratio of the source used as a fertilizer influenced this B coefficient. So not only the soil, but also the type of addition influences the response of the soil to phosphorus fertilization intended as soil phosphorus balance. Another way to use the nutrient balance is not to calculate the nutrient requirements of crops, but to use to calculate an indicator of the system's sustainability. So if we calculate an annual balance is with a simplified formula that only includes the additions of fertilizers or manure, and as the only negative term, the crop removal with yield, well, we have a simplified formula, a simplified balance formula. And this is can be used as a sustainability indicator, because if this balance is greater than zero, it means that the system is accumulating the nutrient, or that the system is losing the nutrient, so there are losses from the system. If the balance is instead negative, well, it means that the crop is taking up more nutrients than supplied with fertilizers, so and the crop is depleting the soil reserves. Here we applied this approach of sustainability indicator to three systems that were maize for silage and timely harvested maize, to the double crop of maize for silage and ryegrass, and to the rotation of maize for silage and grassland. And I will show you the average of 8 years. Okay, we calculated here the accumulation of the nitrogen balance across years. And the nitrogen balance, as you can see, was negative in all the systems. It means that all the three systems had a negative balance. Negative balance means that all the systems were not fertilized enough and were depleting the reserves of the soil. But the nitrogen that was used by the crop, taken up by the crop, didn't come from fertilizers, but came from other sources. And this was true for all the three systems, apart from only one case here of the double cropping system. And for both levels of fertilization, there were 170 kilograms of nitrogen and 250 kilograms of nitrogen in all systems, both regardless the type of fertilizer, urea, farmyard manure, or slurry. So where did this extra nitrogen come from? We have seen the answer before, while we're looking at the mineralization of soil organic matter. And in fact, if we observe the change in soil organic matter content in only these years, 8 years of this observation, well, there was a reduction in the soil organic matter content. It means that the crop depleted the soil reserves to take up the nitrogen, all the nitrogen that it could use. Finally, we can apply the mass balance not only in the place that we know best, so the long-term experiment, but also in other cases. And I will show you very briefly the effect of calculating nitrogen balance in some private farms. Again, we have a system with an input and an output, but in this case, we have to simplify the entries that we can quantify. In particular, only few entries can be quantified in this field mass balance calculated in private farms, because some are not known and some are negligible, such as irrigation water in our cases does not bring a lot of nitrogen, but also seeds that do not contribute a lot to this balance. And as an only output, we can calculate the crop uptake, while all the rest is included in the difference, so in the system balance. So this simplified balance that was calculated in the crops in different type of farms. You can see differentiated in different type of farms, because this paper that was published about 20 years ago had as an objective the comparison between different systems of livestock systems. In particular, we compare the field nutrient balance and the field nitrogen balance that we obtained in the dairy cows farms with the one that we obtained in farms that rear beef for meat production, with another type of bovine farms that is typical here in Piemonte, that is for meat production, but the type of rearing is different, and so it's a meat production, but the management of bovines is different. And we also compare it with pig farms. As you can see, we calculated the field scale balance, and we have a total input, the total output as removal, and the crop balance. The crop balance was positive in all type of farms, but the typical Piedmontese way of rearing animals, so the suckling cows system, yielded a crop balance that was positive, but less positive than the other type of farms. Where did this extra nitrogen come from? Well, it came mainly from manuring. So manuring was even higher than year removal in all cases. It means that farmers didn't have any idea of how much nitrogen they were distributing when using their manures. And they also distributed mineral fertilizers to their crops. It means, again, that they could save a lot of mineral fertilizers because there was an excess, even if only manures were used. Where does this, so the excess of nitrogen measured at the field scale is due to an excess of manure. If we scale up and consider instead not a field balance, but a farm balance, so it's a farm gate balance, and it's another mass balance that is calculated using the farm gate as the limit, as the boundary of the system. It means that everything that comes into the farm is an input. Everything that comes out of the farm passing from the gate is an output. As inputs, we have the mineral fertilizers, but also live animals, the bedding materials, the feed, atmospheric deposition, nitrogen fixation. While as output of the farm gate balance, we have the animal products and the crop yield that is exported outside the farm, and manures that are exported outside the farm. While everything that is recycled within the farm is not considered in this kind of approach. So again, we have a farm balance that is highly positive for all types of farms, with a better situation in some situations rather than others. And here we can see where that excess of nitrogen in manure comes from. It comes from feed. Feed is the main source of nitrogen that enters the farm, and it will end up in excess of nitrogen in the manure. So we have, if we scale up, we see that the problem of excess of nitrogen It comes from feeding, how we feed the animals, as we rely too much on the feed that is produced outside the farm itself. Where does this come from? Well, we can scale up again and see the results of this is instead a paper that we did not contribute to. It's a paper that calculated the nitrogen balance based on into this region and came out of the region based on data that nowadays are rather old, as they are 20 years old. So the problem of having too much nitrogen as feed comes from the fact that we import too much nitrogen from other countries, in particular from Northern America and Southern America, and I can tell you that most of it is used as feed. Most of it is maize and soybean used as feed. So scaling up the mass balance can help us understand what is the main problem that we should face to improve the quality of our soils and our water in Europe. So the nutrient balance is a good approach that can be used for several purposes and can be used through this process scaling up to evidence the key causes of the imbalance that we observe in our soil. So I thank you for your attention, and if you have questions, I'm here to answer.
Thank you so much, Laura. And yeah, if anybody has any questions.
One question. Laura, thanks a lot for this fruitful presentation. My question is, in your long-term experiments, have you observed any physical structure changes? The physical structure change of the soil? Since I think from your balance calculation, for instance, the changes of the soil bulk density may influence those calculation process, in my opinion.
Yes, you're right. Some changes in the soil bulk density, but we have not yet explored the something like the aggregate stability. That's something that we would like to explore. It is a soil that is not prone to produce a lot of big aggregates. The aggregates are quite small, so it's possible that we will observe that based on the fact that we are increasing the soil organic matter content in some and we expect to see a change in that. In a case, it is so also calcium carbonate is an important source of formation of aggregates. This helps the formation of aggregates apart from soil organic matter. In any case, thank you for your suggestion, and I think it's a very good suggestion to increase our database regarding long-term experiment. And another thing that I would like to do in the next future is to make properties of the soils. It's something that we didn't, but now, well, all the hypotheses that you have formulated can be right. Unfortunately, we could not understand why, because in this meta-analysis, we collected long-term experiments, and we tried to understand which were the main factors for the changes that we observed. But it was, in particular, it was not easy to have significant results putting everything together. Despite the number of long-term experiments we have in Europe is rather high, because 80 is not is a good number. Well, they are not enough to really understand which are the main factors affecting this different response. And in particular, we should maybe go into the details of single long-term experiments to explain why, which are the processes involved in that. It could also be, yes, a sum of different factors. Despite the statistical model that we use, try to separate the effect of soil from the one of climate. There could also be some overlapping.
Okay. And I also have another question. You showed, for example, in TUdi, the different types of different fertilization. Is the tillage system always the same, or are there also differences? I guess it's because I was always thinking if there are different tillage systems, are the effects of the different fertilization systems also varying? Yeah.
Yes, you're right. No, at Tufrati we have the same system of incorporating residues. There's not really plowing, because the plots are too small for plowing, as the soil would move from one plot to another. So we use a spading machine that incorporates and remixes the soil at 25, 30 centimeters depth.
Mhm. Thank you very much.
Thank you.
Any other questions before we move to the next lecture? Okay, I think then we can proceed. Yeah, so our next lecturer, Professor Kunju, is a professor at the School of Land Science and Technology, China Agricultural University. He graduated from University of Science and Technology of China in 2006, majoring in chemistry and receiving his MS degree in agronomy from Seoul National University in 2010, and then later his PhD degree from the Department of Soil and Environment at the University of Copenhagen in Denmark in 2014. In 2014, he received his PhD in soil environmental sciences from the University of Copenhagen, where he also worked as a research assistant in the Department of Plant and Environmental Sciences. He joined the School of Land Science and Technology, College of Resources and Environment of China Agricultural University in October of 2016 and has been working there since then. So a little bit about his research. His major research focuses focus has been on soil biological fertility and the influence of utilizing agricultural and urban waste as fertilizer. With the exponential growth of intensified animal production, animal manure is becoming a major waste product, requiring safe, sustainable, and sanitary management. Urban waste management approaches are also undergoing rapid changes, which have particular impacts on nutrient cycling opportunities in many countries. When waste is utilized on arable land, its fertilizer value and effects on soil or the environment should be optimized, and negative effects of xenobiotics and pathogens on ecosystem and human health should be eliminated. His recent research focus has been thus broadened to also cover these areas, namely utilization of agricultural and urban waste as fertilizer, woodland application of animal manures and other waste products from agriculture, industry, and society, and their fertilizer value and effects on social quality, gas emissions, and nutrient losses to the environment, as well as microscale dynamics of soil process, investigation of hotspots and complex media via novel techniques such as planar apoptose, diffuse gradient, and thin films, and others. And today, he will be presenting on the topic of the impacts of soil organic amendments on greenhouse gas emissions. Thank you so much for being here, and yeah.
Yes, thanks a lot for those nice introductions. And I will try to share my screen. Just a minute. Yes. Thanks a lot, everyone. My topic for today is greenhouse gas emissions after those application of organic amendments in the soil. Uh, since the main topic is the greenhouse gases, actually we know that three major gases, the carbon dioxide, methane, and the nitrous oxide, those are the main contributor to the global warming. And the nitrous oxide is a key role in my presentation. Actually, nitrous oxide, it plays several important roles in different industries. Like in medical science, nitrous oxide is known as the laughing gas, which is used as anesthetic mixtures in medical treatment. Of course, the nitrous oxide can also be used as a kind of oxidizer to promote the combustion of the energy carrier. And most importantly, nitrous oxide is a very strong greenhouse gas. It has a much higher global warming potential than the carbon dioxide. And right after the first industrial revolution, the nitrous oxide concentration in the atmosphere is sharply increased. Actually, it's around from 200. Right now, the concentration of nitrous oxide in the atmosphere is around 340 ppb, which is much higher than before the 18th century. Since the nitrous oxide is a key issue for the global warming or for the greenhouse gases, so we need to know where do those processes can produce the nitrous oxide. In the agricultural soil, actually, mainly there are two processes to produce the nitrous oxide. One is the nitrification. That's under the oxygen conditions. There are plenty of oxygen. The nitrification process will produce the nitrous oxide as a byproduct. While under the oxygen limited conditions, the denitrification will occur, and nitrous oxide is one of the intermediate products in the denitrification process. As the nitrification and denitrification process, of course, they need the nitrogen substrate, like the nitrogen fertilizer and animal manure will produce, will supply plenty of the ammonium and nitrate for the nitrification and denitrification. Therefore, the amendments of those nitrogen fertilizer or animal manure will cause a risk for the nitrous oxide emissions. Uh, One of the studies have observed that almost around 10% of the nitrogen will be emitted as the nitrous oxide under the pig slurry applications. So actually, the soil organic amendment application do have some very high risk to produce the nitrous oxide emissions. Those amendments, those organic amendments will produce the different carbon source and the nitrogen source, and the different environmental variables and the cropping systems will could produce to influence the nitrous oxide emissions. Once there is nitrous oxide emission, it will definitely increase the greenhouse effect and at the same time reduce the fertilizer use efficiencies. Therefore, we do have some different measurement, or we have different strategies try to mitigate the nitrous oxide emission. One of the practice nowadays, like the no tillage as one of the conservation tillage, it's applied to increase the soil quality and to increase the crop yield as well. Since the no tillage, it can improve soil physical structure and also increase the soil organic matter. Therefore, the no tillage practice is quite popular to improve soil quality. At the same time, actually, we want to ask, since the no tillage practice, they have the straw cover that remains the straw on the surface of the soil. That means it will provide some extra carbon source for the soil. Can this no-till practice is also beneficial for the nitrous oxide mitigation. We want to answer those questions, then we conduct those long-term experiments. In this long-term experiment, it was established 2011. That is more than 12 years. It is in the Lishu experimental station in the northeast of China. And the main treatment is including two kinds of treatment. One is the conventional tillage, the other is no tillage. And the plot size is more than 1,000 square meters per plot. In this long-term experiment, actually we conduct around two kinds of treatments. One treatment is different of tillage practice, like a conventional tillage. The second one is the long-term no tillage practice. It's more than 12 years. The third treatment is no tillage within 2 years. It's kind of short-term practice. And the crop we grow is maize. This crop is kind of row planting crop. So we have different in the row scale. We have different rows. One of the row is cropping rows with the maize plant. Another kind of row is the interrow, where there's no plant there. And our fertilizers actually were placed in the cropping rows, and our crop residue in the no-tillage practice, the crop residue were placed in the interrow, while in the conventional tillage, the crop residue was removed. That means there's no crop residue, there's no straw was placed in any rows. Under this experiment, we try to monitor the soil pH and the soil DOC content. The DOC is the dissolved organic carbon. In the whole cropping seasons, we have observed that for the pH content, for the soil pH, we have observed the long-term no-tillage practice actually reduced the soil pH. The long-term no-tillage practice is the blue and the yellow bars, which almost has the lowest pH in all the time. Such pH decrease probably is due to the crop residue degradation. In the degradation of crop residue, it will produce some organic acids. Those organic acids are one of the reasons to reduce the soil pH. At the same time, actually, the long-term no-tillage practice could increase the soil dissolved organic carbon content. It's also due to the degradation of crop residue, since they have more carbon source to input to the soil. Well, for the soil mineral nitrogen, for the nitrogen availability, we monitor the ammonium content and the nitrate content. And we also observed that the soil mineral nitrogen, particularly for the ammonium nitrogen, which was much higher in the long-term no-tillage practice. And those increase of the ammonium content probably is due to the higher soil organic matter and from the crop residue degradation, and also because some of the nitrogen supply is much stronger in the long-term no-tillage practice. And of course, we also monitored the whole season for the nitrous oxide emissions in the field. And we found that for the nitrous oxide emissions, the hot moment of the emissions occurred in the rainy season. It's from the July to August, where the most of the precipitation is occurred in this period. That means most of precipitation event will induce the nitrous oxide emission. And between among the different treatments, the no-tillage practice actually have increased the nitrous oxide emissions. For the cumulative emissions although there are not that significant difference, but we have observed for the long-term no-tillage practice actually could potentially increase the nitrous oxide emission risk. We found that those higher nitrous oxide emissions uh, quite associated with the lower soil pH and higher nitrogen availability. The lower pH actually they have kind of effect, have some effect to inhibit the nitrous oxide reductase. That means the lower pH will reduce the nitrous oxide reduction to the dinitrogen gas. That means if under the lower pH conditions, the nitrous oxide will be not easy to be reduced to nitrogen gas. Therefore, the nitrous oxide emission will be higher under lower pH conditions. So in this field trial, we found that the cropping rows will induce higher nitrous oxide emissions due to the fertilizers which were placed in the cropping rows. That means the fertilizer will provide more nitrogen substrate, more nitrogen availability for the nitrous oxide productions. And the long-term no-tillage practice covered with the straw, actually, since it can reduce the soil pH and increase the nitrogen availability. Therefore, it could increase the risk of the nitrous oxide emissions. And those two conclusions, including the cropping rows, including the straw cover in the soil surface, that means it's kind of increase the soil heterogeneity. We want to understand are those soil practice induce more hotspots of the nitrous oxide. Then we focus more on the interface between the soil and the soil organic amendments. Since most of the amendments is including the manure or the fertilizers, those amendments are usually applied as particles into the soil. So the amended particle, the interface between the amended particle and the soil have the gradient effect, like they have a different physical structure of different chemical gradients, and also have different biological functions. One of the important chemical gradients or chemical parameters is the soil oxygen. The soil oxygen actually will determine the microbial activity, also the nutrient availability. Therefore, the soil oxygen is one of the key factors influencing the production process of the nitrous oxide. The challenge is it's very difficult to directly measure the soil oxygen content in a small scale, since the soil is quite heterogeneous, and also soil oxygen is very sensitive to the soil disturbance. If we use any sensor or any probe inserted to the soil, then will disturb the soil structure and also influence the soil oxygen content. So recently, we developed the plant optode. It's a membrane sensor, which is kind of a non-invasive technology, can continuously measure the soil oxygen content inside. Here is the system we used for the plant optode system monitoring the soil oxygen content. We place the optode membrane. This is the membrane sensor to inside the soil chamber, which is directly contact with the soil. And we use LED as the emission light, as the excitation light. Under this light, this membrane will emit the fluorescence light. The emitted light intensity is depend on the soil oxygen content. And those emitted light will be recorded by the camera. Things like if the soil oxygen is 0%, it's lower content, then we get very dark images. If the soil oxygen is 100%, is much more higher, then we get very bright images. Under this system, we can see in the laboratory, we have this system to monitor the soil oxygen content, to continuously monitor the soil oxygen content. We have this is the soil chamber, and we have the LED light to excitate the membrane signals. And those optical signals will be recorded by the camera, and finally will process those camera recorded images to get the soil oxygen content in situ. Here we designed a laboratory incubation experiment. We apply pig manure as the soil organic management. We have two categories of the treatment. One category is only place the pig manure in the soil. And the pig manure was specially distributed in the soil. In the control treatment, there's no pig manure, and in the mixed treatment, we mix the pig manure homogeneously in the soil. In the layer treatment, we place the pig manure as a concentrated layer in the center of the soil. And for the patch treatment, we place the pig manure in the center of the soil as a patch. And for the next category of the treatment, we have the same spatial distribution of the pig manure. The only difference is we add ammonium sulfate as a nitrogen fertilizer in the soil. Here is we monitor the soil oxygen dynamics. We will take two treatments for examples. In the patch treatment, we place the manure as a patch in the center of the soil. Then in the center of soil is a manure patch. You can see how much oxygen is consumed around the patch area. And here is the oxygen scale bar. That means the darker, in the dark color, that is lower oxygen content. In the bright color, means have more oxygen content. And for the patch treatment, you can clearly see that the oxygen was depleted in the manure patch and then radically extended to the surrounding area. After around 3 days, after 3 days, only the four corners, which is further away from the manure patch, still kept at the oxic conditions. Well, in the patch treatment with the ammonium sulfate, the oxygen depletion is much faster. The oxygen depletion is expanded to the whole soil area. After 3 days, actually only the surface layer around 2 to 3 millimeters, those soil are still kept a little bit oxic conditions since because of the oxygen can be diffused from the contacted air. So here is all of the treatments. We can clearly see that in the mixed treatment, since the manure and the soil are homogeneously contacted together, therefore they have the highest or have the fastest oxygen consumption rate. And after adding the ammonium sulfate and this nitrogen fertilizers application will also enhance the oxygen consumption. That's mostly because the manure has an anaerobic degradation that consume the oxygen. Also, the ammonium nitrification is also a process which consumes the oxygen. Therefore, once we have the manure and the nitrogen together, then the oxygen is depleted very fast. With the nitrous oxide emission monitoring, we can see that in the mixed treatment, since the oxygen is consumed fast, therefore they have the fastest nitrous oxide emissions and also the earliest emission peak, followed by the layer treatment and the patch treatment at the slowest oxygen depletion, therefore have the slowest nitrous oxide emission. In all of the treatment with mineral nitrogen, that with ammonium sulfate applications, those nitrous oxide emissions is getting higher, and the both emission and the emission packs are much higher than those treatment without mineral nitrogen. So we have concluded that in this system, we have found that the oxygen availabilities determine when the nitrous oxide will be emitted. That means the oxygen availability will regulate the temporal patterns of nitrous oxide emission, while the carbon and nitrogen availability will determine how much of the nitrous oxide will be emitted. That means the nitrogen and the carbon availability will determine the magnitude of the nitrous oxide emissions. Except the soil oxygen, another important factor is the soil pH. The soil pH actually will determine the microbial activity as well as the nutrient availability and also the plant growth as well. And since the oxygen is important, soil pH is also important, we want to analyze the micro scale heterogeneity of the soil amendment interfaces. We want to try to monitor the pH gradients and the oxygen gradients in the soil amendment interfaces using the plant optical system. That in that way we can explore the nitrous oxide hot spots and hot moments as well. In this experimental trial, we place the straw and the biochar in the center of the soil. The biochar is produced from the pyrolysis of the straw under 500 degrees. And those amendments just placed as a patch in the center of the soil. And we also use the plant optical system to monitor the soil pH dynamics. For the straw treatment, we place the straw in the center of the soil, and we can see the pH dynamics around the soil area. Here is under this dark blue means lower pH, and this bright color means the higher pH. Here we can want to see the dynamics of the pH between the soil and the straw interface. In the center, actually, the straw will produce some organic acid under the straw degradation, therefore the lower the soil pH. And after several days, those pH, after several days, the pH is the lower pH will get recovered since the produced organic acid will be consumed by the microbes afterwards. Therefore, those pH decline will get recovered after several days. Well, for the biochar treatment, the biochar actually contains a lot of alkali content. That means the biochar will increase the soil pH. And this increase actually is very localized conditions. The biochars will release some alkali content is close to the biochar area. And this kind of phenomenon will keep for several days. and we analyze the pH spatial distribution around the straw patch. For the temporal dynamics, the straw addition around the straw patch actually will lower soil pH around 10 hours. After 10 hours, it reaches to the lowest point. And then after 1 day, there will be gradually those organic acid produced from straw degradation will be consumed by microbes again, and the soil pH will be get recovered to the original level. And for the biochar treatment, actually the soil pH is in the patch treatment in the center. The pH will increase more than 1 unit, that's around 8.5. And during the experimental period, the biochar actually have very stable pH around the biochar patch. And once away from the biochar patch, for instance, at 20 millimeter away from the biochar patch, that is, the soil actually will not almost will not affected by the biochar application. Once it getting closer to the biochar patch, the soil is slightly or around more than 0.5 unit of pH increase. That means the biochar application actually have very limited influencing zones. They could increase the soil pH, but this area is very limited to the surrounding area. It cannot be expanded to much larger area. At the same time, we also monitor the gradients, the soil oxygen gradients in the interface between the organic amendments, like the straw. Since the straw was placed in the center of the soil, we can see how much oxygen was depleted around the straw area. Actually, for the straw, we found that the straw in the center of the straw, the soil oxygen content was not reduced. Only the interface, that means the adjacent area of the soil and the straw, the contact area of the straw and the soil have plenty of activities to reduce the soil oxygen. You can see that it's like a ring of the soil depletion zones. Those depletion zones are developed as a ring around the straw patch. And in the center of the straw patch, the soil oxygen content was not depleted much. And we have considered that since the straw has much more porous structures than the soil, therefore those kind of porous structures are beneficial for the oxygen diffusion. So the straw degradation will consume oxygen, but at the same time, the porous structure of the straw are beneficial for the oxygen diffusion. That means beneficial for the oxygen input. So the balance between the oxygen input and the oxygen consumption, this kind of balance cause this kind of the ring zone of the oxygen depletion patterns. But for the straw-derived biochar, the biochar is quite inert. That means the biochar are not easy to be degraded. Therefore, the biochar degradation could be negligible. And the oxygen content in the biochar patch also very high. It's also because of the biochar have a lot of the porous structure, which is also beneficial for the oxygen diffusion. Therefore, the oxygen in the biochar patch are not reduced. Yes. For the next one, this is the temporal dynamics of the soil oxygen content in a different treatment. For the straw treatment, we consider straw have dual roles. One of the role is the straw contains a lot of level of carbon which will be degraded by the soil microbes and to consume the oxygen. And on the other side, the straw have very porous structures, which is beneficial for the air diffusion. That means the oxygen diffusion. Therefore, they have some different temporal patterns for the straw amendment. For the biochar treatment, the biochar carbon are quite difficult to be degraded. Therefore, the biochar actually not consume any oxygen. And at the same time, the biochar have very porous structure, which is beneficial for the oxygen diffusion. Therefore, in the biochar area, the soil oxygen is the soil could be kept as oxic conditions. We monitor the soil pH dynamics, and also the nitrous oxide emissions. And we found that in the straw treatment, the soil pH was negatively correlated with the nitrous oxide emission rate. As previous, I mentioned that nitrous oxide could be reduced to be the nitrogen gas. But under the lower pH conditions, those nitrous oxide reduction will be inhibited. Therefore, the lower pH actually was beneficial for the nitrous oxide emission. And for the biochar, the higher pH conditions inhibit the nitrous oxide productions. And we also to correlate the soil oxygen content between with the nitrous oxide emissions. We found that the average oxygen content is also negatively correlated with the soil nitrous oxide emissions. Just under the very lower oxygen content, you have observed very high nitrous oxide emissions. So therefore, we conclude that those kind of nitrous oxide probably they are produced from the denitrification process. And the higher oxygen content, we observe very little nitrous oxide. That means the nitrification process will probably not produce much nitrous oxide. So for my talk, here's my summary. We have concluded that the macro-scale heterogeneity of the soil oxygen content and the pH dynamics is quite important for the nitrous oxide emissions. In the interface of the soil manure system, the oxygen dynamics regulate the temporal patterns of nitrous oxide emission. And for the soil straw system, the straw could at the same time to increase the soil oxygen consumption, and at the same time also increase the soil oxygen diffusion. For the soil biochar system, the biochar could cause localized alkalization and also can enhance the soil aeration and keep the soil oxic. Therefore, the interfacial microscopic heterogeneity could directly influence the nitrogen turnover, and particularly for the nitrous oxide emissions. Yes, that's all of my presentation. Thanks a lot for your attention.
Thank you so much. And yeah, once again, the floor is open if anybody has any questions.
I have one.
Yes.
Thank you very much for your very interesting speech. I found it very impressive. So, in particular, the second part regarding the device that you have to measure oxygen concentration, so that you can go deep to study the processes, the microscale. And have you ever tried it with the plants on? In particular, I was thinking of the rice plant because of its ability to move oxygen from the leaves to the roots, and that could also change the rhizosphere, and so it could be related to this. To the production of N2O, but also the device could also be used for other different kind of studies.
Yes, thanks a lot for this comment. Actually, we have collaborated with other rice plant groups. Right now, we are conducting the rice box with the rice planting in the rice box, and we want to monitor those oxygen contents around the rhizosphere, and we want to monitor those kind of to establish any links between the oxygen content and also for the methane emissions. In this time, for the rice plant, probably the methane emission is more important than the nitrous oxide. Yes, it's quite important. Thanks.
And I have also another small question regarding instead the first part of the one regarding the field experiment. And as you
Yes.
said that rainfall increased the N2O emissions. And I was
Yes.
wondering if that was because rainfall was a lot, and so there was more anoxic conditions for the soil, or if it was instead due to the fact that rainfall stimulated microbial transformations. So depending on how much water was brought by the rainfall, what is your interpretation?
In our case, in that year, that was 2023, that's 2 years ago. Actually, those precipitation actually is not that heavy. That means the soil water content is increased, but it's not strictly anaerobic conditions. So probably under those kind of rainfall events, the the soil is not oxygen depleted, but is oxygen kind of limited. In that case, it promote the nitrous oxide emissions. Of course, if under the strictly oxygen depletion conditions, probably the denatrification process will produce nitrogen gas, not nitrous oxide. That's our interpretation.
Thank you very much.
Thanks.
I also
Yeah.
have a question. I
Yes.
did not understand. For example, with the planar uptode, did you investigate disturbed samples or undisturbed soil samples? Because I was wondering if you can just use this methods to understand the processes better, or do you really um, investigate an undisturbed soil and can derive the process directly at the field with that?
Oh, well, right now in the presentation, we use the repacted soil. It's not the undisturbed soil.
Yeah.
And in the currently, we are trying to monitor we apply this plant optode in the undisturbed soil. We have tried to apply those kind of this system in the field, but in the field that's a lot of disturbance. It's not the soil conditions. That's a lot of even including a lot of the animals. There's it causes a lot of interference in those optical signals. Anyway, we are trying to kind of establish this planar opto system in the field to monitor the soil oxygen content in the field and to monitor soil pH content in the field as well. It may take several. I think it take time.
Yeah. And I was wondering, um, because in terms of conservation agriculture, it is not a good result that no tillage results in higher N2O emissions. Are there any, um, could we still try to go towards conservation agriculture and have some measures to deal with the higher N2O emissions? Um,
I know there's uh, uh, have found other studies actually I found that those kind of no-TD practice sometimes can reduce the nitrous oxide emissions due to they have changed the microbial activities. And in our studies, actually, the nitrous oxide emissions will getting higher in the no-TD practice. And right now, I don't know exactly what's the main factors causing those differences. And therefore, we are trying to. This is the first year studies. We haven't get those. We we are trying to monitor those greenhouse gases, particularly for the nitrous oxide emissions, for um, continued for this year and the last year. And we are thinking to collect more data and get more solid conclusions. Anyway, this is only for 1 year observation, so we cannot get very solid conclusions yet.
I understand.
Therefore, we want to continue this experiment in the future.
Yeah.
Yeah.
Thank you very much.
Thanks.
Are there any other questions?
Hey, hello.
Hi.
Hey, hello. Six. Yes. Laura Kunzhu. So thank you very much for this. It's a last training course. Thanks,
Yes.
Gonza. Thanks, Gonza. Where are you? You're south of
Thanks.
China. No, back to Austria.
You're muted, Günther. Günther? You're muted.
You're muted.
Cannot see,
No,
cannot hear you.
sorry.
Okay,
No, I
that's
did
good.
not contribute much to, thank you, Laura and Kunshu and Biljana for moderating. I did not contribute anything to this. So thank you all to that. And personally, I'm back in Vienna, in Austria. So last week in China was a nice experience. Thank you.
Good.
Yes. Thank you, Biljana.
Yes, yes.
Yeah, thank you. Yeah. Yes, I'm pleasure to organize this type of training. It's very valuable to the student to learn a lot from a different field. Yes, I'm glad to see you here and know you.
Yes.
all from Europe. I hope you can come to China and welcome you all. Laura. Yeah.
Yes, thank you so
Good,
much
good.
to everyone who participated. Thank you, of course, to our lecturers and for taking the time to prepare these lectures and give them. And yeah, it's been a pleasure. So thank you all for being here today. And if there are no other questions,
Yeah.
okay.
Yeah.
All right. Then thank you so much to everyone who made the time again.
Thanks a lot.
Thank you.
Thanks for the
Thank you.
organizations.
Thank you. Wishing you a lovely day and evening.
Bye-bye.
Thanks.
Bye.
Bye bye.
Bye.